the basics?
Yes.
Yeah. Yes, that continue.
George John Mendel. He was the
It's
father.
Gregor. It's Gregor.
Sorry. Gregor
Yeah, you can
John
say
Mendel.
G.J. Mendel or just
G.J.
Mendel is
Mendel.
fine. Yeah.
Okay. He is known as the father of genetics, as he studied on the inheritance of the traits, and he used the plant Pisum satium, that is pea plant. He conducted artificial pollination, cross-pollination experiments using several true breeding varieties. Those were having very contrasting traits. Okay.
Right.
Okay. Now, why he chose the pea plant?
Yes, that is the question.
Yeah.
Why he chose pea plant?
Because the pea plant is like it is an annual plant with like short life cycle.
Yes, right.
So several generations can be studied within this small period of time. Then it have like bisexual flowers. So I think it is a property. It is a bisexual flower. Okay.
Yeah, and it is cleistogamous.
Yeah, cleistogamous.
So what is the advantage of this? Because
this helps in pollination. Pollination as
which pollination?
Yeah, this pollination. Okay.
Both self and cross.
Self and cross.
It's on the board. Can you see the board? Can you see the board?
Yes.
Yeah.
Like predominantly they choose self pollination, but several times they take cross pollination as because
they do not take it. They all the time pea plant does self pollination because it is a closed flower. Okay, it never opens, but it is possible to open this flower by hand. Like you can make it open and remove one part, either the male or the female, and this way you can allow this plant to only undergo cross pollination.
Yes.
Okay, so this manipulation is very easy in pea plant. That's why Mendel chose this.
Okay, then the pea plant produce a large number of seeds in one generation. Pea plant can be easily raised and maintained and handled as the number of no. There are many contrasting traits. As we know, there are 14 contrasting characters and traits.
There are 7 traits. There are 7 characters. For each character there are 2 contrasting traits. Okay, so 7 characters and 14 traits, or 14 varieties of plant that Mendel used. Okay.
Yes.
Yeah. Tell me about them.
Some traits. Okay.
Tell me about the character and the traits. Tell me the character name, the dominant trait of it, and the recessive trait of it, which is on the board.
Okay. First, I will take like character, that is seed shape. Seed shaped are having 2 traits, that is dominant and recessive. One dominant one is round and recessive one is wrinkled. Then I will take seed color. In seed color, we have yellow as dominant and green as recessive. Character flower color. In flower color, we have dominant violet color, recessive white color. Then pod shape, full like you can say inflated dominant trait, and contrastic
constricted. Yeah,
it's like similar to constricted. Recessive trait. Then we have pod color. Pod color is green, dominant. Yellow is recessive. Flower position is axial, dominant, and recessive is terminal one. Then stem height is tall is dominant. Then short dwarf is recessive. Okay. So there are 7 pairs of contrasting traits of pea plants selected by Mendel.
Yes. Then in the last, I will take from here. So in the last class, I told you about the 1 gene inheritance and 2 gene inheritance. Basically, there are 3 laws of Mendel that we study. The first is called law of dominance, then law of segregation, and law of independent assortment. I ask you to go back and read Punnett Square, Genotypic Ratio, and Phenotypic Ratio as a homework. Did you do that?
Yes.
Yes, very good. So this will make today's class easier for you to understand. Now, today we will start with individual laws. Okay, so write down today's topic: laws of inheritance given by Mendel. Okay, or based on Mendel's work. Laws of inheritance. Okay, so the first law that we're going to start with is, of course, law of dominance. Okay, now as the name suggests, law of dominance, so which means this law states, let's first define it. This law states that in a heterozygous condition, one allele is dominant, or one allele is expressed over the other allele. For a particular character. Okay. Now, the allele that is expressed is called dominant allele. And the allele which is not expressed, or allele which remains hidden or silent, or not expressed, is called recessive allele. Is it clear?
Yes.
Okay. Now let me tell you what do we mean by allele. So as I told you, let's take an example of one character. Tell me any character that we should start with. Let's start with height. Okay.
Yeah.
So height is a character. Now it has 2 alleles. One is capital T, right?
Yes.
And the other is small t. These are the 2 alleles for this character, which gives rise to 2 traits. Now, what are alleles? You know what are alleles already? Alleles are alternate versions of the same gene. Okay.
Yes.
So basically the gene is same, but there are certain changes in that, and that's one allele gives rise to different phenotype and the other allele gives rise to slightly different phenotype. Now, on the basis of these alleles, in a gamete, there can only be one allele, but when both the gametes fuse to make zygote, 2 alleles for each character is there in our body, right? So if in our body, capital T and capital T is there, or if in pea plant's cell, capital T and capital T is there, then it will give rise to tall. Okay. And this is called pure line tall. Okay. Or
yes,
homozygous tall. Is it clear? Basically,
yes.
this is dominant. Both are dominant. So it's also known as homozygous dominant. Is it clear, the first scenario?
Yes.
In the second scenario, it can be capital T with a small t, two different alleles. Now, interestingly, this is also tall, but this is called a hybrid tall because there are two types of alleles mixing here, or it is also known as heterozygous tall. Homo and hetero means homo means same, similar, both are same type, same alleles. Hetero means different, both are different alleles. Now in this case, we see that capital T is dominant over small t. Yes or no? In the when both are in a heterozygous state, this is what this definition says. In a heterozygous condition, like in this condition, one allele expresses over the other. So what's happening here is that this allele is dominant, right?
Yes.
And is being expressed. And this allele is recessive and is not expressing itself. It is hidden. Is it clear?
Yes.
Expressed and hidden. So only in a heterozygous condition you get to know which one is dominant and which one is recessive. The third condition is in which both the recessive gametes are present together. So this plant will give rise to a dwarf plant, and this is called pure line dwarf or short, and it always will be homozygous. If it is recessive, it always will be a homozygous recessive. It cannot be a heterozygous recessive. You understand? Because in a heterozygous condition, the dominant one expresses and the recessive one is basically hidden. Is it clear?
Yes.
Now, how did he figure out law of dominance? Let's try to reason that. So what Mendel did is, Mendel started with a tall plant. Okay. Pure line tall. What do I mean by pure line tall?
Whose ancestors are like, whose parents are heterozygous?
Not about the parents. Pure line tall is whose genotype is capital T, capital T. Okay. And it was crossed with a short or a dwarf plant. And dwarf always has to be pure line. You know that because it's a recessive character. So there is no need to tell a dwarf is a pure line or heterozygous. It always will be small t and small t, right? Now this is the parent generation. You understand? Now, when you make the cross in the F1 generation, you saw that all the plants were capital T and small t. All. You know how it happened? It happened like this. So this phenotype can give rise to two gametes, right? One containing capital T and the other containing capital T. This will also give rise to two gametes, small t and small t. So if I make a Punnett square, this is called a Punnett square, where basically on one side, I keep one tall parent's gametes, capital T and capital T. You understand?
Yes.
And on the other side, I keep other parents' gametes, small t and small t. And when I make a cross, all the kids will be capital T, small t, capital T, small t, capital T, small t, and capital T, small t. So all of them are basically tall, right? But hybrid tall. So he saw that all the progenies were tall in F1 generation. Now this is interesting, no? Because you started with tall and dwarf. So you were expecting that some kids should be tall and some kids should be dwarf. Yes or no?
Yes.
Some will be like the parent A and some will be like the parent B. But instead, everyone was like parent A, tall. So he got confused. What's happening? And he realized that this allele for dwarfness is hidden. It's not expressing itself in the first generation. Makes sense to you, Zed?
Yes.
So he thought, how can we make this recessive allele expressive? Will it express itself? So he self-crossed the F1. Okay. Self-crossing F1 means reproducing F1 among themselves. So when you self-cross F1, basically what you are doing is you are crossing capital T small t with capital T small t. This is F1, right? As a result, F2 will be produced now to get F2. So what happens is in F2. Now tell me what kind of gametes will be made here, parent A.
So heterozygous tall, then homozygous.
Gametes are single, right?
Yeah.
Capital T and small t. How will capital T will form? There is no second capital T, right?
Yes.
There will be only two gametes, capital T small t, and here also capital T small t. We are currently talking about just one character, tallness. So gamete will be single. Okay. So we will make a Punnett square now. Again, let's keep parents A genes here and B here. So the gametes are capital T and small t, and here also it's capital T and small t, right?
Yes.
What will be the first kid?
Capital T.
Perfect.
Capital T, small t.
Okay.
Capital T, small t.
Okay.
Small t.
Yes. Now, do you see something here? There are three different types of genotypes. One is this, two belong to this, and one is this. So in F2 generation, this is your F2. Okay. In F2 generation, there comes a concept of genotypic ratio, and phenotypic ratio. There was no such ratio in F1 generation because everyone in F1 was capital T small t, all. Okay. But here, do you see that this genotype is different? So there are three genotypes possible: capital T, capital T small t, and small t small t. This is 1, this is 2, and this is 1. You understand? 1, yes. 2, and 1. So the genotypic ratio becomes 1:2:1. And look at the phenotypic ratio now. How many of them are tall?
3.
Yes, these 3 are tall, right?
Yes.
And this one is dwarf. Yes or no?
Yes.
But what were the parents? Both the parents were
homozygous.
Both the parents were tall. Heterozygous tall.
Heterozygous.
Now, interesting thing is, look up. When we took a tall and a dwarf parent, we got all tall. But when we took 2 tall parents, we got a dwarf. Now, this tells us that in F2 generation, there is a homozygous recessive condition possible. Then only a dwarf can come in, right? So the phenotypic ratio can be tall or dwarf. Genotypic means about the genotype, the genetics. Phenotypic means about the visible characters. The word pheno, write down. The word pheno is about visible characters in biology. The science, the stream of biology that is called phenology, deals with the visible study of visible characters. Is it clear?
Yes.
So how many are tall?
3 are tall.
3 are tall and 1 is dwarf. So the genotypic ratio is 1:2:1. And the phenotypic ratio is
3:1.
3:1 in the F2 generation. And this kind of cross is called a monohybrid cross. What it is called?
Monohybrid cross.
Yes. Why monohybrid? Because only one character is studied. Please make this.
Okay.
Let me know. I'll scroll back when you're done this. So we are starting with parents, P, generation, then F1 and F2. We are going till F2. Is this done? Shall I scroll down?
Yes, yes.
Yes, then self cross F1.
So done.
Done?
Yes.
So is the law of dominance clear to you?
Yes.
Yeah, and the monohybrid cross is also clear. The genotypic and phenotypic ratio is also clear.
Yes.
Yeah. Now, following law of dominance, we always have, when it's capital T and capital small t, which is when we have a heterozygous condition, we always have the dominant character visible. Now this law, write down, law of dominance is not a universal law. Which means it does not hold true everywhere, and Mendel did not know about this. Okay, so it's not a universal law, and which means it has exceptions in nature. Okay.
Yes.
Now we are studying about exceptions of law of dominance. Now exceptions could be, if something is showing dominance, then exception could be that thing is not able to show dominance, right? Obviously. Now first exception is law of, no, not law of, it's not a law. It is, first exception is incomplete dominance. Okay.
Yes.
Now what do you understand by incomplete dominance? In this, write down, in incomplete dominance, one allele is not able, one allele is not able to express itself fully. So no one allele is able to express itself completely over the other allele in a heterozygous condition. Is it okay?
Yes.
Yeah. Now this can happen because of three reasons. Either this heterozygous allele is not that efficient in the presence of the other allele, or is it not non-functional allele. Now, an example of this is the flower color. The flower color of snapdragon plant. Okay, just remember this example. Flower color of.
So it is known as 4 o'clock. Yeah?
No, 4 o'clock is Mirabilis jalapa. Okay. Snapdragon is not 4 o'clock. Okay, 4 o'clock is Mirabilis jalapa. This is called and the scientific name is Anterinum.
Anterinum majus.
Yes, Anterinum species. Okay, so this is the plant. Now in this plant, let me first tell you what happens in a snapdragon plant. There are three possibilities. Okay, for the character flower color. So the character is flower color. There are two alleles. Let me delete this. There are two traits. One trait is red color flower, which is the dominant trait. Is it clear? Just like the pea plant, there were traits which were dominant and recessive. You understand? So one is red, which is dominant trait, and the other is white flower, which is a recessive trait. Do you understand, Seth?
Yes.
Now for red, if it is homozygous dominant, capital R, then it is in a homozygous dominant condition, it gives red. Okay, and it gives white in a homozygous recessive condition, because in both these two conditions there is no concept of dominancy, right? These both are dominant, these both are recessive, these two are recessive, correct?
Yes.
An interesting thing happens in a heterozygous condition. So what should be, if normally following the law of dominance, what should be the color of this heterozygous condition?
It should be pink.
Following the law of dominance.
Red. It should be red.
Yes. Following the law of dominance, it should be red in a heterozygous condition, right? But here, this gives rise to pink flowers. So do you see blending in of the characters happening? There is blending of the character. If you take a red color and mix it with a white color, which color will come up?
Red and white. Pink.
Yes, you mix red and white. Have you done painting?
Yes.
If you mix red and white, it will form pink.
Red and white, pink, yellow, and something.
Yes. So how are these colors formed? So red is basically a pigment, right?
Yes.
Which is giving the flower a red color. White is also a pigment which is giving the flower a white color. These two pigments are blending here because no one pigment is dominant over the other. Do you understand?
Yes.
So it is called pink flower. So here, write down, in this heterozygous condition, there is blending of traits. So blending of traits happen. Is it clear?
Yes.
And this basically is called incomplete dominance. There is no dominance. There is no complete dominance. Okay?
Yes.
First note this down, please.
Settle.
Done?
Yes.
Okay, now let's go to the genetics of it. So let's start with the red flower. Cross to a white flower. Okay, so this is the parent's generation. Okay, in the
Yes.
F1, what will happen? Tell me. If we make a Punnett square. Yeah.
In F1, one gamete
What should I write?
from R. Okay.
What should I write here?
R and R.
Which R?
Capital R, smaller.
Why smaller? It is for this parent.
For white, yeah.
No, but this is parent A and this is parent B.
Yes.
On one side, we write the gametes of parent A. The gametes will be capital R and capital R. Here, the gametes will be
smaller.
Yes. Now tell me what will be the offsprings?
Capital R, smaller.
All four offsprings in F1 will be heterozygous. And what will be the color of the flower?
Pink.
Yes, all will be pink. You understand?
Yes.
Now this is interesting, no? Now if you in F1 all will be pink. Now if you self cross F1, can you do this, Seth, and tell me
yes.
what will happen? So basically I'm asking you to do this cross. Please make the gametes, make the cross in which both the parents are pink to begin with.
So one will like phenotypically.
No, don't do it like this. Make a Punnett square, make a cross, and tell me, you have to tell me two things. In the F2 generation, you have to tell me the genotypic ratio and the phenotypic ratio. Okay? Do it. This is the question on your screen. Take 2 minutes. I'll be back. Okay? I'll get some water. Okay, Zed?
Yes.
Okay, done, Zaid?
Yes, yes.
Yes, so let's make the Punnett square quickly, and you will tell me what will be the gametes. So what are the gametes for parent A?
For parent
and
A,
parent, yes. So I'm
both are
writing. What should?
So R, capital R.
Yes.
Small r.
Okay.
And
for
capital
parent.
R.
Okay.
Capital R is small.
Okay. Tell me, the first child will be?
First will be R, capital R, capital R. Second will be capital R, small r. Third will be capital R, small r.
Okay.
And last will be smaller, smaller.
Perfect. Now tell me, what different genotypes are present? Either it can be capital R, or it can be capital R, small r, or it can be small r, right?
Yes.
So what is the genotypic ratio?
1:2:1.
Is it same like when there was law of dominance being followed?
No, no, sir, no.
Look again.
Was 1:3.
What was the ratio when law of dominance was being followed?
It's same.
It's same, right?
Yeah.
Now, what will be phenotypic ratio? How many types of phenotypes will be there now?
3 types. 3 types.
What are the 3 types? What are the 3 types?
First will be red.
Okay.
Second will be pink.
Okay.
Then white.
If there was law of dominance, it would have been either red or white, no pink, right?
Yes.
So what will be the phenotypic ratio?
1:2:1.
So 1 is red, 2 are pink, and 1 is white. So this only is red, right?
Yes.
These 2 are pink, and this one is white. Yes. Right? Yes. Now, is this phenotypic ratio same as before?
Yes. Phenotypic ratio is different. That was 3:1.
Yes. So in incomplete dominance, the phenotypic ratio changes, right?
Yes.
But the genotypic ratio remains the same. And this is an F2. Now make this. Done this, right? You already made this. Yeah. Now write down in incomplete dominance. In incomplete dominance, the phenotypic ratio is affected. But the genotypic ratio remains the same. Is it clear? Yes. So this is law of not law. This is incomplete dominance, which is an exception of law of dominance. Okay. Yes. Yeah. Now let's go to the next exception. There is another exception of law of dominance known as codominance. What does the word codominance tells you, Zed? Co means co-education, cooperation. What is co-education? Co-ed school.
Together, something like that. Together.
Yes. Yeah. Yes, right. It means together. Perfect. So write down in codominance. In codominance, both the alleles in a heterozygous condition are expressed equally. In other words, they co-express together. Okay, which means that there is no recessive allele, right?
Yes.
Both are codominant. Is it clear?
Yes.
Now, an example of codominance is in AB blood group. Okay, so in humans, what is the blood group in humans?
There are 4 majorly. A.
The system is called ABO blood group or blood grouping system. So in this blood grouping system, let's start with A blood group. So there are 3 possibilities. Sorry, there are 4 possibilities. First is A group, right? To have a A blood group, what are the chances? So let me first tell you, in this grouping system, it is called multi-allelic system. Okay, what it is called? Multi-allelic or polyallelic. Do you understand? There are more than 2 alleles here.
Yes.
Now the first allele is IA. This codes for antigen A. Okay. The second allele is IB, which codes for antigen B. And the third allele is small I allele, which does not codes for any antigen. So no antigen is produced because no sugar is produced. Basically, these antigens are sugars, right? So no sugar produced. So these are the 3 alleles of the same gene. Do you understand?
Yes.
Now to have a blood group, the possibilities are either you have IA from one parent and also IA from other parent. In this case, you are homozygous for A and your blood group will be A. The other possibility is you have IA from one parent and small I or IO from the other parent. Do you understand? O means no antigen. Is it clear?
Yes.
Yeah.
Yeah.
Same is for B group. So to have a, now here this A is dominant over O, of course, because A is a antigen and O is no antigen, absence of any antigen. So if something is absent, the allele is present, but that allele is not producing any antigen. So of course A will be dominant over O, right? Yes. So here law of dominance is being followed in A group. In B group also, either it is IB or it is IBIO. Yes, 2 possibilities. Yes. Third is for O group. O group means a recessive group. So the only possibility is IO, no other possibility. In all these 3, there is no codominance. There is normal law of dominance followed. Codominance is always only in this 4th group, which is called AB group. To make a AB group, there is only one possibility. IA. So from one parent you got A antigen or IA allele, or and from other parent you got B antigen through IB allele. Now both these 2 alleles are codominant. Is it clear?
Yes.
So both produce their sugars, and as a result, both antigen A and antigen B are present on the surface of RBC. Write down. So this group, specifically fourth group, shows codominance. Okay.
Yes.
Please make this chart.
So finished.
Done?
Yes.
Also, you understood?
Yes.
Now let's do some questions. What kind of questions from this blood grouping and codominancy comes in exam? Okay. There is another example of codominancy in the pea plant only. Okay. Mendel did not know about codominancy also because the type of characters he studied were visible characters. If Mendel would have studied the character for the starch granules, which was not possible, like he did not study it at that time, but if he would have studied that, he would have known that his law of dominance is not followed there. Okay, but let's do some questions on AB blood group first. So I'm giving you questions, you will solve it. Okay, write down the question. If a woman with blood group O, if a woman with blood group O marries a man with blood group AB, okay, this is the parents. What is the possibility of blood groups in the progenies in F1? Tell me the phenotypic ratio and the genotypic ratio. You understood?
Yes.
Okay. So make the cross and let me know. This is the question. Okay, solve it. Take 2 minutes. Try to be fast and also accurate. Okay, Zed?
Yes.
Yeah, I'm just here. Let me know when you're done.
So can I say?
You're done?
Yes.
Okay, very good. Now let's do it together. Tell me, first thing, what is the genotype of the woman?
IO, that is, we can write small I.
Yeah, let's write it in IO. IO and
IO.
IO. And what is the genotype of the father?
IA, IB.
Okay. Now tell me, if you make a Punnett square, what should I write as gametes here for the female?
So we can write IO, IO.
IO, IO. And for the male?
IA, IB.
IA, IB. Yes, what will be the first child?
IA, IO.
Okay.
IA, IB, IO.
Okay.
Again, IA, IO. IB,
Okay.
IO.
Okay. What will be the phenotypic ratio? Let's first come to the genotypic ratio. What are the two genotypes possible?
So there are two genotypes, that is I think IAIO and IBIO.
So what will be the genotypic ratio?
2:2.
2:2. And what will be the phenotypic ratio?
Again 2:2.
Yes, because these two will have blood group A. Yes. And these two will have blood group B. So 2:2. The second part of the question is, what kind of dominancy is there in the parents and in the offsprings? So in the parents, there is a codominancy, right?
Yes.
But in the kids, in the offsprings, is there any codominancy? Is there any codominancy?
No.
So what is there? Complete dominance. Okay.
Yes.
You understood? Very good. Well done. So let me give you one more question, which is actually from the, let me just pull it out for you, the question. Okay. Just wait. Yes. I'm giving you one question from the NEET papers. Okay, Zed?
Okay.
I hope it opens up. Okay, I'll dictate you the question. Okay.
Okay.
Okay, it opened. Yes, this is question number 23. This was asked in 2015. We paste it here. Can you see the question? Make it a little big. So you have to solve this question and tell me the answer. You have 2 minutes. Okay, Zed?
Yes.
It's a new question. Read the question carefully and tell me which one is the right option, and then we'll figure out why. Let me know when you're done.
So can I tell the answers?
Can you what?
Yes, sir. Can I tell the answer?
Yes, you're done.
Yes, sir. Option number A.
Yeah. Option number A.
Option number A.
Okay. Now, have you also figured it out why that is the answer? Yes. Okay. So help me with that. Let's go to. Let me choose the pen. Okay. So tell me. A man with blood group A marries a woman with blood group B. What are all the possible blood groups of the offsprings? Now, for the man, what is the genotype?
IA, IO.
IA, IO? Can't it be IA, IA?
Yes, yes.
Right? Because it's not telling you whether the man is homozygous or heterozygous. You understand?
Yes.
Though your answer A is correct, but we have to consider both now. And let's say the mother is
IA, IB, IO. IB,
IO and
IB.
Now, taking this one, you also have to take this one as consideration. If you take this one and cross, then all the offsprings will be of the AB blood group, right?
Yes.
Because if these two are crossed, it will be like AB blood group for all offsprings. You understood how?
Yes. Only AB blood group will come.
Yes. Because if we make the Punnett square, for one parent it will be IA, IA, and for the other it will be IB, IB. So all will be IA, IB, IA, IB, IA, IB, and IA, IB. Am I correct?
Yes. So this can we say like codominance?
Yes. So there will be codominance. Yeah. But if you cross these two, let's say if these two possibilities are there, because this is also blood group A and B, then case will be different. Then you will know the exact right answer. So by chance, you actually took the right one to do the cross. So in this case, the gamete will be IAIO and IBIO, right? So this kid will be of the AB blood group, IAIB, codominance. This kid will have A blood group. This kid will have blood group B, and this kid will have blood group O. So all 4 blood groups are possible. Do you understand?
Yes.
That's why the right option is option A, B, AB, and O. Okay? But if just to confuse and to make this question a little harder, what they can do is they can say a man having blood group A or homozygous, a man with blood group A, homozygous or heterozygous. If they're right, then you have to figure out through this. This is homozygous and this is heterozygous, right? This is heterozygous A. This is homozygous A. Yes or no?
Yes.
This is heterozygous B and homozygous B, if these terms are used. I hope you understood.
Yes.
Okay. Very good. Now, there is one more question that have been asked. Now, let me dictate you that question. Write down that question. A child has blood group O. A child has blood group O. You're writing? Is that?
Yes.
Yes, a child has blood group O. If the father has blood group A, if the father is A and mother is B, then what will be the genotypes of the parents? What will be the genotypes of the parents and the possible genotypes of other offsprings? So this child's siblings. Written the question?
Yes.
Yes, figure it out.
So I think same answer will be there. The father and mother will have heterozygous combination of genes.
Yes.
So we will have 4 possibilities of blood groups. That will be AB, A, B, and O.
Yes. So indirectly, it is the same question. This question which you have written has come in board, but it is basically asking you the same thing, right?
Yes.
This is what I wanted to ask. Perfect. So this same thing is asked. If a father is A and the mother is B or vice versa, and you have a O, so they give you this information, then they give you this information, and they give you this information, and they ask, what about the others? You understand?
Yes.
Yeah. So it's easy, right?
Yes.
Okay, very good. So this was all about what they can ask for the blood group, and there are many other questions. Please keep practicing those questions. And now we come to the second law of Mendel. So write down second law of inheritance. The second law is called.
Can I take 2 minutes break?
Yes, please.
1 minute break. Okay.
Yeah, sure. I will also just connect my laptop. Okay. Yes, you're back.
Yes.
Yeah. Let's go to the second law, which is law of segregation. So law of dominance and its two exceptions are clear to you: codominance and incomplete dominance.
Yes.
Now the second law is called law of segregation. It is also known as commonly as law of purity of gametes. Is it clear?
Yes.
Now this is the only universal law. Okay. So it is a universal law. And what does it mean? It has no exceptions. This law is followed everywhere in biology without any exceptions.
Okay.
So clearly, if there is no exceptions, this law must be stating something very, very fundamental in biology.
Yes.
Yeah. So let's write what it states. Write down. Law of segregation states that law of segregation states that while making gametes or during gamete formation, each gamete receives during gamete formation, each gamete receives one allele of the gene, or we can say one copy of the gene. Okay, allele is basically a copy, right? Yes. One allele of the gene selected randomly. Okay. Yes. Or write down, in other words, you can also state it like this. During the gamete formation, each allele segregates independently of the other allele in a gene. Okay.
Yes.
So which means there is no blending. There is no blending of alleles during gamete formation. So all these sentences say the same thing. Okay.
Yes.
And this law has no exceptions because we have been following this law all throughout. Even in the law of dominance, do you see that when this was the offsprings, like these were the parents, we segregated their alleles separately?
Yes.
Every time we made the alleles, even when we were doing law of dominance, or let's say in codominance also, when we were making this cross, we segregated the alleles separately, right? These two alleles got separated independently of each other to make gametes, right? Same happened here also. This capital R got independently segregated from this small r. It was a heterozygous gene, but both the copies, both the alleles got separately segregated to make gametes without any blending, right?
Yes.
Yes. So that's why law of segregation has no exceptions. So we do not have to study any exception of it. It is simple law. It's called law of purity of gametes, and it's done. Written? Note down.
Yes, okay.
Done?
Yes.
Now, as I already told you in the beginning, that the first law, law of dominance, and the second law, law of segregation, talks about one gene interaction. Okay, they come from the inheritance of one single gene. The third law, which is called law of independent assortment, let's start with the third law. Write down, law of independent assortment. This is based on independent. This is based on a dihybrid cross. Okay.
Yes.
Now, what is a dihybrid cross? Where we talk about the inheritance of two genes. So before going into law of independent assortment, I'll discuss with you 2-gene inheritance. Okay.
Okay.
So what is 2-gene inheritance? This is basically when we are talking about 2 genes inherited together. And normally in biology, multiple genes happen like to inherit together, right? It's not like each gene goes single, right? In our every chromosome, we have hundreds, thousands of genes, right? They are all inheriting together. Yeah. So this is the actual scenario. Now this this law says the same thing about genes, what law of segregation was saying about alleles. Okay. Law of segregation was saying that 2 alleles present in one gene segregate independently of each other to make gametes, right?
Yes.
Law of independent assortment says that two genes, and each gene will be having 2 alleles now. So 2 genes segregate independent of each other for during gamete formation or during inheritance. Okay. Now, when Mendel said this, Mendel did not know about linkage and recombination. Okay. Now let's understand 2-gene inheritance first. Tell me any 2 genes to begin with, 2 characters of pea plant.
So we can take height again, and we can take
height and
seed shape, maybe.
Okay. Okay. Seed shape. So as you say, we are now talking about the inheritance of height and seed shape together. Okay. So let's see what happens. Okay. So you said height and seed shape. So let's cross for height, a pure line, capital T, capital T. This is pure line tall. Okay.
Yes.
And seed shape, what is the dominant seed shape? Round.
Yes.
Yes.
We can take
capital R, capital R. So what is this plant? Tall and round. Pure tall, pure round. And let's cross it with dwarf and wrinkled. Okay.
Yes.
Now this is parent generation. So tell me what will happen in F1. So for that we have to make gametes. So you tell me what are the different type of gametes that parent A will make and parent B will make.
So it will be T, capital
Now
T,
remember,
capital R.
Yes. Now remember, in the gametes there will be at least 1 allele from each gene. Okay. Yes. So only 1 allele goes in the gamete, but if there are 2 genes, then from each gene 1 allele will go. So you said capital T and capital R, right? Is there any other possibility?
No.
No, right? And similarly for this parent, what is the gametes?
Small t, small r.
Small t, small r. So when they will cross, what will be the F1?
So
all will be capital T, small t, capital R, small r, right?
Yes.
So what is the phenotype? All are hybrid tall and hybrid round. Yes.
Yes.
So F1, all the plants are tall and round. We started with tall and round and dwarf and wrinkled, and you see that in next generation only tall and round character is visible, but this is hybrid tall and hybrid round. This we can only know from the genotype, not from the phenotype, because hybrid tall looks exactly the same like a pure line tall. Tall is tall, whether it is hybrid or pure line, right?
Yes.
But the interesting thing is, so there is no genotypic, no phenotypic ratio because everyone is tall. Now you tell me, what will happen if self cross of F1 is done? So basically,
what should I say?
Yes, this cross made with this cross. What will happen? So this is F1. What will be the F2 like? Tell me the genotypic and phenotypic ratio.
So first we'll write T, capital T, capital T. Then
okay. Let's do one thing.
Okay.
Let's write the gametes first. So what are the possibilities here now? What will be the gametes for parent A?
Capital T, capital T R.
Capital T?
Capital R, capital R.
Sorry, capital T?
Capital T, capital R.
Okay. This is one gamete.
Okay. Capital T, small r.
Okay. This is another gamete.
Then small t, capital R, small t.
Okay. This is one gamete.
Then small t, small r.
Okay, these are the 4 gametes. So the same 4 gametes will be for this also because they are both the same parents, right?
Yes.
Now, can you make a Punnett square and tell me what will be the genotypic and phenotypic ratio?
Yes.
Yes, quickly. Let me know when you're done, okay? Seth?
So total 16 squares are coming, correct?
Sorry?
So total 16 squares are coming in the Punnett square. So
yes.
Is it correct? Yeah.
Okay. Yes, that's correct. Because on one side there are 4 gametes now, and on the other side also there are 4 gametes.
So can I tell the phenotypic ratio?
Yes, please.
First time it is genotypic. Okay. It's coming 1:2, 2:2, 2:2. Okay. 1:2.
Yes, continue.
1:2, 2:2.
Okay, let me also make a Punnett square with you.
Yes.
Okay, then I'll be able to explain better. So now there are 4 gametes. Correct?
Yes.
Now, tell me the gametes now. Capital T, capital R.
Small
capital T, small r.
Small t, capital R.
Small t, capital R. Small t, small r. Same is here. Capital T, capital R. Capital T, small r. Small t, capital R, and small t, small r. Now the first one be capital T, capital T, capital R, capital R.
Yes.
Capital T, capital R, small r. Capital T, small t, capital R, capital R. Capital T, small t. Keep checking, okay?
Yes.
Keep cross checking. Capital T, capital R, small r. Capital T, small r, small r. Capital T, small t, capital R, small r. Capital T, small t, small r, small r. I hope you're checking, right?
Yes.
Am I correct?
Yes, yes.
Yes. Now look here. A short trick is to go diagonals, okay? Go through diagonal, go diagonally. You will see this is a unique phenotype, right?
Yes.
Yeah. So we will write in phenotypic ratio 1 is to
2.
These two on the diagonal are same, right?
Yes.
To 2. There is no other like this. Then this is unique.
Unique.
To 1. Then these two are same, right?
Yes.
Is to 2.
Then 4. 4 in the diagonal.
Yes. Then all these 4 on the diagonal are same, correct?
Yes.
Here is to 4.
Then in the center.
Then again these two are same, right?
Same. Yes.
Is to 2.
Then
this
again
one is
center.
different. Is to 1.
Then 2.
And these two are same. Is to 2. And this one is different. Is to 1. Okay. So it becomes 1:2 is to 1:2. So it first increases 1 2 1 2 4 2 1 2 1. Okay.
Yes.
Now tell me the genotypic ratio. Now what are the possible genotypes? Because there are 2 genes. First tell me what are the possible genotypes. Either it can be tall and round, or it can be tall and wrinkled, right? Or it can be dwarf and round, or it can be dwarf and wrinkled, right?
Yes.
Yeah. So tell me, what are how many of them are tall and round?
So tall and round are 9.
Yes.
1. Okay. And
tall and wrinkled
and dwarf are wrinkled. Sorry, dwarf and wrinkled are 3.
Okay.
Okay. Wrinkled. Sorry. Dwarf and round. Sorry. Dwarf and round are 3 again.
Okay.
And dwarf and wrinkled are is 1.
Perfect. So you see that the genotypic ratio is 9:3:3:1.
Yes.
Right? And a phenotypic ratio is this for a dihybrid cross F2 generation. Okay. Is it clear?
Yes.
Will you remember this?
Yes.
Yeah. Note this down.
Sir, already finished.
Done. Okay. Now, do you see that this tall and round and dwarf and wrinkled are actually parental genotypes? Parents were tall and round and dwarf and wrinkled to begin with, right? So these two are parental combinations. What are they?
Parental combination.
These two are parental combinations. But in any of the stage, was any of the parent tall and wrinkled? Was any parent tall and wrinkled?
Yes.
No.
No.
Yeah. No parent was tall and wrinkled anywhere.
No.
Progenies are tall and wrinkled.
Yes.
Was any parent dwarf and round?
No.
No. So these two are non-parental combinations. Yes?
Yes.
Now, out of total 16 progenies, so total progenies are 16. What is the percentage of parental combination and non-parental combination? Tell me.
Sir, how do you know that percentage?
How do you calculate percentage?
Percentage divided by. Okay, non-parental upon parent.
Percentage is for parental combinations. How many are parental? 9 plus 1, 10 are parental, right?
Yes.
10 out of how many?
9.
16.
Sorry.
Into 100. This is the way to find. Tell me, what is the parental combination? This is how you find percentage for your exams, no? Marks obtained over total marks into 100.
Yes.
Yeah. Tell me.
So 62.5.
Exactly. And what is the non-parental recombination?
It will be 6 by 16 into 100.
What did you say? 62.5.
Yes, yes.
What is the non-parental combination percentage?
So approximately 67.5.
What?
67.5.
6 over 16 into 100.
So 37.
Yes. 37.5. Right?
Yes.
Now remember this.
Or otherwise we can just subtract it and we can get it here.
Yeah, exactly.
Yeah.
But you did not do it, so I thought,
yes.
I'll tell you after that. 1% you find, the other one can you just because total percent maximum can be 100, right?
Yes.
So 37%, around 37% progenies here are non-parental, right? Which means none of the parent had those character combinations which these progenies got.
Yes.
Do you now understand how sexual reproduction causes variations?
Yes.
We studied this in chapter 3 that in chapter 1st, we started with, you said a statement, asexually reproducing organisms have no variation and sexually reproducing organisms which makes gametes and fuses gametes have variation. This is that variation. No parent to begin with was tall and wrinkled, but now we find progenies, 37% of the progenies which are either tall or wrinkled and dwarf and round, non-parental, right?
Yes.
Yes. So this is, so remember this ratio because this ratio will come up again, but you understood how to do a dihybrid cross? Yes. And what is a 2-gene inheritance?
Yes.
Yes. So let's quickly define, let me define, write down. Law of independent assortment. Write down. This law states that the alleles of the law states that the alleles of 2 or more different genes gets sorted into gametes independently of one another. Okay. Oops. Which means one gene does not influence or alleles of one gene does not influence alleles of the other gene. Alleles of the other gene during segregation. Is it clear? Yes. So this is what law of independent assortment is. And if you pay attention, it is saying the same kind of thing for 2 different genes, what law of segregation was saying for 2 different alleles in the same gene. Yes?
Yes.
So when you say that for two different alleles, but in the same gene, that is universally true. That is always followed. But when you say that two genes, no gene influence the other gene, this is not universally true. So write down, this law is not universally accepted. Which means in biology, it has exceptions. Right?
Yes.
So it has exceptions. Now, when Mendel said this, he did not know of the exceptions, okay, because he only worked in pea plant. So he could not figure out what are the different exceptions. Now, for law of independent assortment, the exceptions are, do you know what are the exceptions?
So test cross?
No. Test cross is not an exception. Test cross, I'll tell you.
Sorry.
Exceptions is linkage between genes. So genes are linked. That's why they actually do not segregate independently. So linkage between the genes is an exception for law of independent assortment. Is it clear?
Yes.
Now let's define it first. I'll explain both these
so is
things.
there one more like recombination also?
Yes, recombination happens when there is no linkage. Okay. Recombination is a normal phenomena that happens during meiosis. But if there is linkage, recombination does not happen.
Yes.
Okay. So today I'll define linkage and I'll define its relationship with recombination. In the next class, which is on Thursday, we will discuss linkage and recombination, test cross and back cross in detail. Test cross and back cross has nothing to do with linkage or recombination. That is a test to figure out the genotype. Okay.
Yes.
Yes. Now write down linkage. Two genes having a physical association. Two genes having a physical association due to close proximity on a chromosome. Which leads to failure of segregation of the two genes. Okay. Now this is called linkage.
Okay.
Simply linkage means, so basically what happens is, you know, on a chromosome. So if I make a chromosome, you know that chromosome looks something like this, right?
Yes.
Let's say there is a gene A on chromosome. Okay. So before explaining, I'll make a chromosome as one. Like there is only one copy of chromosome, let's say. The other part is not made yet. This is a chromosome, okay? And there is a gene. Oh, sorry. Use something. There is a gene called gene A. Okay. And there is another gene called gene B. And then there is another gene called gene C. Okay. These are genes. So you see that A and B are very closely present together.
Yes.
Let me make it even more closer. Oh. How can I just delete? Yes. Let me make it even more closer to each other. Can you see now? They are very, very close.
Yes.
Yeah. Now, due to their very close proximity, and between A and C, there is a long gap, and between B and C also there is a long gap, right?
Yes.
But between A and B, there is very small gap. Now, most of the times when chromosomes make recombinations, okay, recombination is actually the physical event where the chromosome breaks and joins back to its homologous part. Now, all this I will explain to you in the next class through diagram. But just know, if the genes are very, very close together, the chances of them separating is very less. So for A and B, there is a linkage between the two because both of them are very, very close. So there is a high linkage. Okay. And between A and C, there is very low linkage or no linkage. Is it clear?
Yes.
And the relationship between linkage and recombination. So recombination give rise to all these different non-parental combinations. Okay. So this which you find 37.5%, write down, it happens due to recombination. Okay. So linkage is inversely proportional to recombination. Is it clear?
Yes.
So what does